Cells are the basic units of life. They are the smallest entities that can be considered living. Some organisms are unicellular, meaning they're just made of one cell. Other organisms are multicellular, meaning they are made of many cells. There are two main categories of cells. We have prokaryotic cells and eukaryotic cells. So prokaryotic cells, the word pro means before, and karyo means kernel, or in this case, we're referring to a part of the cell called the nucleus, the kernel, the interior of the cell. Prokaryotic cells are smaller. They lack the nucleus, hence the name before nucleus, and they don't have these complex membrane-bound organelles, or we could think of that as inside the cell compartments. So their cells are relatively simple. They are what make up the organisms in the domains bacteria and archaea. Eukaryotic cells, on the other hand, eu means true, so they have a true nucleus. Karyo, again, is kernel or nucleus. They are larger in size. They have the membrane-bound nucleus, and they have membrane-bound organelles, so they have complex compartments inside their cells. Organisms in the domain eukarya are made of eukaryotic cells. These include animals, plants, and fungi, as well as a number of single-celled types of eukaryotes. All cells, whether prokaryotic or eukaryotic, have to convert energy from molecules they take in, so remember that energy in bonds, into usable energy for the cell, and that energy is in the form of ATP. In prokaryotes, this conversion takes place in the cytoplasm. That's the inside kind of goo space of the cell. In eukaryotic cells, these energy conversions take place in both the cytoplasm and what we call the mitochondrion. Plural mitochondria, single mitochondrion. And in this picture, this is the mitochondrion. There's many of them. This is just showing a few. They're not to scale either, these pictures, these sketches, but they give you an idea of the basic structure of the mitochondria. And the cytoplasm is all this stuff in here, the non-compartments in the eukaryotic. The mitochondria has inputs and outputs. So one input is oxygen, or O2. This is why we breathe. Our oxygen needs to go into our mitochondrion. We also have food, sometimes in the form of glucose. That's a simple sugar type food compound. Then the glucose is converted into carbon dioxide after processing. The oxygen is converted into water after processing. And the part that's really the point of the mitochondria is making this ATP molecule, or high energy molecule there, and that's an output as well. You can see that it's a lot of folds of the membrane. So there's two membranes. We have the inner membrane and the outer membrane. And then we have lots of fold called cristae or crista, singular. That's all these folds here. And then we have the matrix, which is the inside of the whole mitochondria, and this intermembrane space, which is the space between the inner and outer membranes. And we'll see why that's important. Mitochondria are really dynamic. Sometimes we think of them as these little static features, because we see drawings of them, not real mitochondria. But if you watch, these are labeled green, so all the little green things are mitochondria. And you can see they're moving around. They're actually able to divide in half. They move on filaments in the cell. And so they're actually these dynamic structures making the ATP in the cell. Mitochondria originated by a process called endosymbiosis. The prefix endo means inside or in. And then the symbiosis refers to this mutually beneficial relationship. Okay. What the evidence shows is that a unicellular organism, this is a single cell, we can call it the host cell, engulfed a bacterial cell. Bacterium is singular. So we have this little bacterium, and it gets eaten, basically, which is not unusual. That's how a lot of unicellular organisms get their food, is that they eat other organisms. And so when it was taken inside, the membrane that surrounded it from the eukaryote, and here's the prokaryote bacterium inside. So that ends up with the double membrane here. And eventually, this entity, instead of getting digested in a couple cases of life, it was retained in the cell, a bit beneficial for the cell because it made ATP, and eventually turned into the mitochondrion. And that was allowed the cell to make more ATP, and thus was advantageous. So that's the origins of the mitochondrion. And how do we know this? There's a lot of lines of evidence. One is that double membrane of the mitochondrion. The outer membrane looks a lot like a eukaryotic membrane, a eukaryotic cell membrane. And that folded inner membrane, when you look at its parts and structure, looks like a bacterial cell membrane. Showing the engulfing event there. The second feature is that mitochondria have their own DNA. And it looks like how bacterial DNA is shaped. It's in circular. It has bacterial type looking genes in it. And so that's some evidence that the organism or the mitochondrion used to be its own organism. Besides having its own DNA, it has its own ribosomes. And they can make proteins. That's what ribosomes do. They also look like bacteria type ribosomes. And the fourth feature is that mitochondria can split themselves in half to reproduce. They can divide to make more mitochondria, which is a feature of living organisms, that they can reproduce. And mitochondria do this inside the cell. That's the process of endosymbiosis and how the mitochondrion formed, and the evidence that supports that theory. And just to recall, these mitochondria, why are they important for this discussion in this unit? Because they are energy converters. They convert the energy in food. Most specifically, we talk about glucose, but other foods can go into there too. They convert that energy into energy in the ATP molecule. You'll see in the cellular respiration lecture how that process happens.